Welcome to video series on lipid metabolism. Let's understand metabolism of cholesterol in this video. So this cholesterol metabolism is important because these are the questions that will be asked in university examination. Describe synthesis of cholesterol, synthesis of bile acid, bile salts, functions of bile salts and compounds derived from cholesterol. Apart from this, gallstones, cholelithiasis, cholecystitis are very much important from. Let us try to understand metabolism of cholesterol. Whatever the cholesterol that is present in our diet usually contains around 300 milligram per day. Now this cholesterol is whatever the cholesterol esters we are consuming is acted upon by esterase enzyme releasing the free cholesterol. Apart from that, the hepatocytes also synthesize cholesterol by de novo synthesis of cholesterol. Now this cholesterol is released into small intestine. Here the cholesterol is packed into mixed micelle along with phospholipids and long chain fatty acids. Now these mixed micelle presents this cholesterol, phospholipids and long chain fatty acids uh, along the brush border of the enterocytes, wherein all these macromolecules like cholesterol, phospholipids and long chain fatty acids are released into the intestinal epithelial cells. They are in turn packed into chylomicrons and they are through lymphatics they enter into general circulation. Now the unabsorbed cholesterol is acted upon by the intestinal bacteria which gets converted to cholesterol, coprostanol, and this coprostanol gets excreted in feces. What you need to remember is the absorption of cholesterol requires the presence of a special protein called NPC1L1, that is Niemann-Pick C1 like protein, which is required for absorption of cholesterol from the micelles into the intestinal mucosal cells or the enterocytes. Now we have a special drug called ezetimibe which will inhibit this protein and prevent absorption of cholesterol. Okay, coming to functions of cholesterols and the compounds derived from cholesterol. Now, one of the important function of cholesterol is it from the cholesterol bile acids are derived. Now, these bile acids can be classified as primary and secondary bile acids. Now, these primary bile acids are synthesized from cholesterol in the hepatocytes, and these include cholic acid and chenodeoxycholic acid, and they are conjugated with glycine and they are complexed with sodium and potassium salts to form bile salts. Now these bile salts are released into the duodenum, and these bile salts forms mixed micelles and they are required for absorption of long chain fatty acids, cholesterol and fat soluble vitamins A, D, E, K. Now these primary bile acids are acted upon by the intestinal bacteria which convert them to secondary bile acids, and these undergo enterohepatic circulation, and the remaining part of this secondary bile acid they are excreted in feces. Apart from that, so many other compounds are derived from cholesterol. Now this from this cholesterol, this LDL is a carrier protein of cholesterol from the hepatocytes to peripheral tissues. In the peripheral tissues, especially in the skin, cholesterol is responsible for the formation of vitamin D. In ovary, cholesterol is utilized for the formation of estrogen. In testes, it is required for the formation of testosterone. In the nerves, cholesterol provides the function of insulation. In the adrenal gland, cholesterol is required for the formation of glucocorticoids, especially the cortisol, and the mineralocorticoids, aldosterone. So these are the different compounds which are derived from. Now, coming to biosynthesis of cholesterol, this mainly takes place in hepatocytes. The pathway is partly cytoplasmic and partly mitochondrial. Two molecules of acetyl-CoA condense with each other to form four carbon compound, that is acetoacetyl-CoA. This in turn condense with one more molecule of acetyl-CoA to form six carbon compound, that is beta-hydroxy beta-methyl glutaryl-CoA, also known as HMG-CoA. Now this compound enters into the mitochondria where it gets reduced to form mevalonate, which is again a 6 carbon compound. Now this mevalonate undergoes series of phosphorylation reaction to form 5 phosphomevalonate, 5 pyrophosphomemalonate, 3-phosphopypyrophosphommalonate. This undergoes decarboxylation to form a 5-carbon compound, that is isopentenyl pyrophosphate. So this undergoes isomerization to form dimethyl allyl pyrophosphate, which is also a 5-carbon compound. Now these two condense with each other to form 10-carbon compound, that is geranyl pyrophosphate. Now this in turn condense with one more 5-carbon compound, that is isopentenyl pyrophosphate, to form 15-carbon compound, that is farnesyl pyrophosphate. Two farnesyl pyrophosphate condense with each other with the help of synthase enzyme to form squalene, which is a 30-carbon compound. This undergoes oxidation to form squalene epoxide, which further undergoes cyclization to form lanosterol, which is a 30-carbon compound. So from this lanosterol, 3-carbon compounds are removed to form zymosterol, which gets converted to desmosterol, and finally it gets converted to cholesterol, which is a 727-carbon compound. So one point which you need to remember is desmosterol is mainly present in fetal brain, and this is absent in adult brain. Now this desmosterol reappears in adults whenever they suffer from gliomas, okay, or the. Let us try to understand how is this cholesterol pathway regulated. So the key enzyme is HMG-CoA reductase. So the first mechanism of regulation is allosteric regulation, wherein the end product, that is cholesterol, allosterically inhibits this HMG-CoA reductase so that less amount of cholesterol is synthesized in our body. So this is allosteric regulation. Next one is covalent modification, that is short-term regulation. This is done by phosphorylation and dephosphorylation of HMG-CoA reductase enzyme under the influence of glucagon and insulin. Now glucagon activates adenylate cyclase enzyme, so which causes cyclization of ATP to cyclic AMP. This in turn activates inactive protein kinase to active protein kinase, which will cause phosphorylation of HMG-CoA reductase. So phosphorylated HMG-CoA reductase is inactive in well-fed state. So there is inactive in fasting state. So there is decreased formation of cholesterol whenever we are fasting or starving. So well-fed state is under the influence of insulin, and this will stimulate phosphatase enzyme, which will cause dephosphorylation of the HMG-CoA reductase enzyme, and this dephosphorylated HMG-CoA reductase is active in well-fed state, and so de novo synthesis of cholesterol takes place whenever we are having food or when we have food. So this is about short-term regulation of the covalent modification by glucagon and insulin. The next long-term regulation is done by induction and repression of the key enzyme, that is HMG-CoA reductase. So whenever the cholesterol content is low in our body, so there occurs activation of sterol responding element binding protein, sterol regulatory element binding protein, and this will go and bind to sterol regulatory elements, and this induces the synthesis of HMG-CoA reductase. So there occurs increased cholesterol synthesis. So the contra part happens whenever there is increased cholesterol in our body. So there is no binding of sterol regulatory element binding proteins to the sterol regulatory elements on the DNA, and because of no binding, there is no synthesis of HMG-CoA reductase, so decreased cholesterol synthesis. So this is by induction and repression of the key enzyme, that is HMG-CoA reductase. So there are three different mechanism of regulation: allosteric regulation and covalent modification, which are short-term regulation, and induction and repression of the key enzyme, which is long-term. Coming to formation of bile acids and bile salts. Now bile acids are derived from cholesterol. Now cholesterol is a 27 carbon compound. This is acted upon by hydroxylase enzyme to form cholesterol triol and cholesterol diol. Now from this propionate, a 3-carbon compound is removed. So cholesterol triol gets converted to cholic acid, and cholesterol diol gets converted to quinodeoxycholic acid, and these two are primary bile acids. Okay. Now these two are conjugated with glycine and taurine in the ratio of 3:1. So more of glycine is utilized for conjugation when compared to taurine. So and after conjugation they form glycocholate or taurocholate, glyco or tauroquinodeoxycholate. Now these gets coupled with sodium and potassium salts to form sodium and potassium salts of glycolic acid or taurocolic acid, and sodium and potassium salts of glycoquinodeoxycholic acid or tauroquinodeoxycholic acid, and these get stored in gallbladder, and these are bile salts. Now through common bile duct they enter into the small intestine. So in the intestine they undergo deconjugation with the help of the intestinal bacteria, wherein cholic acid gets converted to deoxycholic acid, and quinodeoxycholic acid gets converted to lithocholic acid, and these are secondary bile acid. Deoxycholic acid and lithocholic acid are secondary bile acid, and most of the secondary bile acid undergo enterohepatic circulation and reaches back liver. Okay, so this enterohepatic circulation is inhibited by a drug called cholestyramine, which is also known as bile acid binding resin, so that these secondary bile acids are not reabsorbed and they get excreted. That is how cholesterol excretion is facilitated by cholestyramine, that is bile acid binding resin. Just to summarize, the primary bile acid, the easy mnemonic to remember is PCC, Priyanka Chopra C. You can remember like that. So P stands for primary bile acid, C stands for cholic acid, and the other C stands for quinodeoxycholic acid. So these are the primary bile acid. Secondary bile acid, the mnemonic to remember is SDL. S stands for secondary bile acid, D stands for deoxycholic acid, and L stands for lithocholic acid. So these are the mnemonics you can remember, memorized mnemonics to memorize primary and secondary bile acids. So the bile salts include sodium and potassium salts of glycocolate and taurocolate, and also sodium and potassium salts of glycoquinodeoxycolate and tauroquinodeoxycholic cholic acid or cholate. So what you need to remember is only the primary bile acids undergo conjugation with glycine and taurine, and not the secondary bile acids. A point to be noted. Okay. Now coming to the clinical significance part, the gallstones. So too much of cholesterol can precipitate as gallstones, and this accounts for 80% of the gallstones are made up of cholesterol. So remaining 20% are pigment stones, and this can be either black or brown pigment stones. So the black pigment stones are usually made up of calcium bilirubinate, commonly seen in hemolytic anemia. Hemolytic anemia is a condition where there is excess breakdown of the RBC, and so this bilirubin along with calcium gets precipitates and forms the pigment type of gallstones. That is black pigment. The brown pigment here, whenever they are usually formed in bile duct, and the composition include the calcium, bilirubin, phosphate, cholesterol. All of this gets precipitated in the bile duct, and they can form brown colored pigment gallstones. Okay, now why we need to worry about these gallstones? These gallstones can cause inflammation of this gallbladder, which is known as cholecystitis, and they can cause obstruction of the bile duct, manifesting as severe biliary colic. It is biliary colic is severe pain in the abdomen, and this can impair the excretion of the bile pigments, and patient can develop severe obstructive jaundice, because pancreatic duct also opens in the second part of the duodenum. So this infection can reflect back onto the pancreas, manifesting as pancreatitis also. Now there are four F which act as risk factors for the development of gallstones, and these risk factors include, the four F include female. So the gallstones formation are more common among female who are in their reproductive age, that is fertile, and usually seen after the age of 40, and among females who are fatty, obese, who have got more than the among whom the BMI is more. So female, fertile, 40, fatty is the mnemonic to remember the risk factors for gallstone form. Coming to the clinical significance part, the reference range of cholesterol, the normally the cholesterol is should be around less than 200 milligram per deciliter. So the borderline is around 200 to 240 milligram per deciliter. So hypercholesteremia is considered when it crosses more than 240 milligram per deciliter. So increased cholesterol is around more than 250 milligram per deciliter. The causes include hypothyroidism. So as you all know, thyroid hormone maintain the basal metabolic rate. So when there is hypofunctioning or decreased functioning of the thyroid gland, so this reduces the cholesterol receptors on the liver. So there is decreased uptake of the cholesterol and decreased breakdown and decreased excretion. So cholesterol levels in blood increases. Obstructive jaundice, again what happens, there is obstruction whenever there is obstruction to the common bile duct, or obstruction because of stone or stricture or carcinoma. So what happens, the cholesterol is not excreted into the intestine, so the blood or the serum cholesterol increases. Uncontrolled diabetes mellitus, what happens is excess glucose gets converted to fat, and excess glucose can also get converted to cholesterol, so increased cholesterol. Nephrotic syndrome is a condition where there is damage to the renal glomerular basement membrane with loss of all low molecular weight proteins. As a compensatory mechanism, there occurs increased synthesis of lipoproteins, especially the LDL, which carries cholesterol. So the cholesterol gets increased in nephrotic. Coming to the decreased cholesterol part, so when the levels are less than 200 milligram per deciliter, so the cholesterol, so it results in hypocholesterolemia. So the causes include thyrotoxicosis. Now, thyrotoxicosis is a condition where characterized by hyperfunctioning of the thyroid gland. So there is increased breakdown of the cholesterol, increased excretion, so the cholesterol content decreases. So anemia is a condition where the hemoglobin is low with a decreased oxygen carrying capacity. As a compensatory mechanism, there occurs erythroid hyperplasia, and this increases cholesterol. So malabsorption, what happens? So most of the biomolecules are not absorbed, and along with the other biomolecules, there is decreased absorption of fatty acids. So decreased synthesis of lipids and cholesterol. Severe liver disease, what happens? So the hepatocytes are damaged, so there is reduced cholesterol synthesis. Apart from that, these are all acquired causes. The inherited hypolipoproteinemia, mainly the familial hypo-beta lipoproteinemia and A-beta lipoproteinemia, where the lipoproteins are decreased, so the cholesterol carrying capacity is also decreased, so there is decrease. So coming to assessment part, rate limiting enzyme in cholesterol synthesis is HMG-CoA reductase, which is required for conversion of HMG-CoA to malonate. Cholesterol synthesis occurs in both cytosol, mainly in cytosol and smooth endoplasmic reticulum. So the early steps occur in cytosol, and later steps occur in endoplasmic reticulum. Sorry, initially I had mentioned about mitochondria. It is not about mitochondria, it is endoplasmic reticulum. So, which vitamins is required for cholesterol biosynthesis? Niacin, because niacin is required for NADPH production. For any DNO synthesis, NADPH is needed, which is derived from niacin. So, immediate precursor of cholesterol is cholesterol. Statins act by inhibiting which enzyme? HMG-CoA reductase. So, statins are wonderful drugs which are mainly prescribed as lipid lowering drugs, and they act by inhibiting HMG-CoA reductase. So the example using atorvastatin, rosuvastatin, so are all statins. So cholesterol is a precursor of all except glucose. So it is precursor of bile acids, steroid hormones like glucocorticoids, adrenocorticoids, estrogen, progesterone, testosterone, vitamin D. Conversion of cholesterol to bile acids occurs in, so cholesterol synthesis is inhibited by glucagon, whereas insulin is anabolic hormone, so it stimulates cholesterol synthesis. So also thyroxine and growth hormone, except glucagon, which is it. Cholesterol cannot be converted to fatty acids, so it can be converted to bile salts, steroid hormones, but fatty acids. So this was all about cholesterol metabolism. So if you had liked the video, please do like, share, and subscribe. Next video will be on ketone body metabolism. Happy learning. Thank you.